let us discuss about cytokinins. One more plant growth promoter. Here cyto means cell, kinins division, kinesis, cell division. In the cell division, in some hormones are participating in the cell division. That's why they are named as cytokinins. Here the cytokinins are mainly here extracted from here different materials. First, the cytokinins were isolated from one fish. It is not natural cytokinin. So, from one fish, that is herring fish. Herring fish sperm DNA. So, herring is one fish. From this fish, sperm DNA is it is isolated and it is autoclaved. So, when it is autoclaved, autoclave is nothing but autoclave is like a pressure cooker. So, high pressure, high temperature is present. So, when the DNA is heated under high pressure and high temperature, so the DNA is producing one here cytokinin hormone, that is kinetin. Here kinetin is it is produced and it is extracted. So here it is not natural. It is not natural. Why? Because when it is autoclaved, when the DNA is autoclaved, then only it is produced. It is not natural hormone, and here it is produced from the sperm DNA. The sperm DNA is here when it is destructed, then only it is produced. Next, here some other cytokinins are also produced or extracted isolated from here different materials. The different materials are corn kernels, here from corn kernels are also here some cytokinins are here isolated or extracted. So the corn seeds, maize plant seeds, so which are in the milky stage, from these seeds some cytokinins are extracted. So these are named as zeatin. Corn scientific name is Zea maize. So from these corn kernels or seeds, the zeatin is extracted. It is also one cytokinin. It is natural. So the natural cytokinin. So it is from one animal sperm DNA. It is not natural. But corn kernel, corn is plant seeds. From these plant seeds, the zeatin is extracted. It is natural. And here from coconut milk also, some natural cytokinins are extracted. So first here the cytokinins were isolated from human urine, and from different animals and here plant materials are also the cytokinins are extracted. So it is kinetin, not natural. Zeatin is natural. From coconut milk also here the cytokinins are extracted or isolated. For the cytokinin synthesis, the precursor is here adenine. Adenine is one nucleotide. So from this adenine, the cytokinins are generally synthesized. So it is the raw material for the synthesis of cytokinin. Next, where these are absorbed, where the cytokinins were absorbed. So generally these cytokinins were isolated, or generally these are produced in the here young fruits and root apices, root apex, root tip portions, and shoot buds also. Shoot buds, so which are in the developing stage. Developing shoot buds and young fruits. And all these portions are in the root apex shoot birds, here apical bird or lateral birds or axillary birds. So here in the developing stage, shoot birds are also producing the cytokinin, and young fruits, small fruits, so unripened fruits also, they are producing the cytokinin hormone. So these are the these are producing generally cytokinins. Next, here the functions of cytokinins. Functions of cytokinins. First one is growth of leaves and shoot. Here the leaf expansion and in the leaf chloroplast, leaves production, leaves expansion. In the leaves chloroplast formation, so generally here the cytokinins are involving in the leaf expansion and chloroplast within the leaves chloroplast formation. So these are involving, and in the shoot growth also, these are generally involving adventitious shoots. So from the adventitious shoot formation, adventitious means generally the shoot is developing from the plumule, but if the shoot is produced from other than the plumule, that is adventitious shoot. Sometimes in the tissue culture technique, here the shoot or the stem develops from the some tissue, not from the plumule. That's why that is adventitious shoot. So in the leaf expansion and chloroplast formation, next adventitious shoot formation. Adventitious shoot formation. So these are involving, the cytokinins are involving in these functions. Next, counteracting apical dominance. So first of all, if we remind the apical dominance, it is the function of auxins. So auxins, they are involving in the apical dominance. Apical dominance means the apical bird is producing the apical bird is producing the auxins, and these auxins are transported to the lateral birds, preventing the lateral bird production or lateral bird growth. So that's why here in some plants, here the auxins are produced here, and they are transported to the axillary birds. So these cannot develop into branches. So it is showing more growth. Apical bud is showing the dominance. So it is a function of auxin, but here counteracting apical dominance. So the cytokinins are preventing the apical bud growth. And lateral buds are branches formation. So the cytokinins are inducing the branches formation. So more number of branches are produced. So opposite to the auxins. So it is preventing the cytokinins are preventing the apical bud growth. So more number of branches are formed. So it is completely opposite to the auxins function here. So that's why it is counteracting the apical dominance. So more branches are produced. The plant becomes bushy. Next, third one is delay senescence. So what is senescence? Here aging process, old age, aging process. So the aging process, it is delayed. So if the aging process is delayed, so due to the here in the aging process, the chlorophyll pigments or the leaves turn into yellow color. So after some time, here the total leaves or flowers, fruits, they fall off. Senescence is delayed, aging process is delayed. So it is called as this effect is called as Richmond-Lang effect. Richmond-Lang effect. The leaf senescence or here fruit senescence, mainly leaf senescence, it is delayed by the cytokinins. It is called as Richmond-Lang effect. Richmond is a scientist name. So here leaf senescence is delayed. Next, in the cell division. So it is the main function of cytokinins. Here cell, here kinesis, division. In the cytokinesis, these are involving, it is the main function of cytokinins. Cell division. Next, what uses of cytokinins? Here overcoming senescence. So generally senescence is delayed. That's why here it is due to the effect of cytokinins, the plants are overcoming the senescence process. Next one is tissue culture. The tissue culture technique, the tissue is cultured on the suitable medium that develops into a new total plant. It is a medium, so containing the nutrients or all food materials, and the suitable conditions, the tissue, some tissue, it is developing into the total plant. So here the shoot formation, stem, leaves, flowers, fruits, the formation, and here root formation, these are induced by some hormones or initiated by some hormones. So mainly here the shoot formation, it is induced by cytokinins. The cytokinins are involving in the shoot formation, so it is called as calogenesis. Here the tissue, it is developing and dividing and forming the callus or undifferentiated tissue. Undifferentiated tissue is present. So from the tissue, how the stem, leaves are produced and root system is produced. Here root system is induced by, the root formation is induced by auxins, and shoot formation is induced by cytokinins. So the shoot formation is called as calogenesis. So these are the functions of physiological effects and are functions and uses of cytokinins.